How's it going, everyone? My name is Jordan.
My name is Elian.
And for our group project, we are doing microorganisms, which is chapters 1 and 2 in the textbook. More specifically, we're doing viruses, fungi, and bacteria. So just a little bit about bacteria really quick. First of all, they're named very similarly to other life forms. Their first name is genus, their last name is species, and then those species can be further subdivided into different categories and variations based on their type. Their types can include, cell morphology, which is their size and shape, colony, DNA, or RNA characteristics. Those are all kind of like distinguishing factors that differentiate bacteria from one another. And there are thousands of them in the world. They're extremely difficult to see under a microscope. They're microscopic. So Dr. Christian Graham developed staining techniques just to make it a little bit easier to identify them. Gram-positive bacteria, last name was named after him, of course, but gram-positive bacteria will have a blue or purple stain, while gram-negative will have either a pink or a red stain. So that kind of helps you distinguish, at least some types of bacteria as well. Another way you can kind of distinguish them is you can inoculate them, which is just placing them on the agar plate in a petri dish. And granted, you have to have the right set amount of conditions, such as nutrients and stuff, so they can grow. But that's another way to distinguish what kind of bacteria type you have. Kind of growing them yourself and seeing what's inside. As far as structure, they are called coccus, bacilli, or spirillus. So, one, you'll have coccus, which is spare, spirillus, which is spiral, and then bacillus, which is kind of like a rod shape. So those are kind of the general structures and shapes of that bacteria come in, but there are many more. These are just the tip of the iceberg. Agents that kill bacteria rather than prevent, their overall growth are going to be bactericidal agents. There's also, sporicidal agents, fungicidal and virucidal, but bactericidal is what you're generally going to want to use to kill and get rid of bacteria. So you kill them by using those agents. You can also use dry heat, or you can use unsaturated chemical vapors from sterilizers. And what that's going to do is the heat is going to actually go in there and it's going to destroy their proteins. And it's going to, more specifically, it's going to break down their RNA and DNA. And it's going to completely eliminate them. So that's one way, or a couple ways you can kill them.
Hi everyone, so I'm going to talk about the scope of microbiology and the infection control behind that as well. So what is microbiology? Microbiology is a study of small forms or small life forms that includes bacteria, yeast, fungi, viruses. Also, it can be subdivided into 4 subcategories. So the first one's going to be bacteriology, which is going to be the study of bacteria. Virology, which is the study of virus. Mycology, which is the study of fungi. And the last one's going to be protozoology, which is the study of protozoa. So in order to know or see how infection control started, so we have to go back at 1546 with Girolamo Fracastro. So he was the one who actually recognized that tiny living things or tiny living organisms are actually the one that causes diseases. So if you heard the word like you catch a disease, that actually came from him. In 1667, Antony van Leeuwenhoek, he was the one who said that it's animalcules is the one that causes the diseases. And guess what? Animalcules, these are bacteria, yeast, and protozoa. In 1800, which is one of the significant period, is the golden age of microbiology. So at this age, boiling water and hand washing actually decreases or kills bacteria. In 1900s, microorganism known as bacteria causes cholera, leprosy, and tuberculosis. And from there on out, we have been adapted to that. The next thing that I want to talk about are viruses. So if you look at right here, there are different parts of viruses. So the different parts are the nucleic acid core, capsid, envelope, and then host cell attachment. So Viruses are smaller than bacteria, and the thing about the viruses is that it needs a host to replicate. And one of the things that we have to consider since working in dentistry and as featured dental hygienists is that one of the diseases is HSV, which is the herpes simplex virus. Also, the life cycle of a virus is important because if you disrupt the life cycle of a virus, then that's basically how you're gonna kill it. So there are actually 6 steps on this, which is absorption, encoding, assembly, penetration, replication, and release. And then the last one is going to be the fungi, which includes the mushroom, molds, and yeast. And in the dentistry, thrush is going to be the number one that's going to be a problem. And you need antifungal medication then. And to recap, we talk about viruses, fungi, and bacteria, and microorganisms.
Yep, and we just want to thank you guys for viewing our presentation. And that's it.
Thank you.
Yep.